Here with Hassan and I'm from Group 4. We are going to present about plant cell and tissue culture media and plant growth regulators. So, what is tissue culture? Tissue culture is a technique of growing new plant tissue by transferring them into an artificial environment in which they can continue to grow and function. So, this plant cell needed medium to carry out this tissue culture procedure. There are many functions of the medium. First, they provide water to the plant cell. They provide mineral nutritional needs, vitamins, growth regulators, access to atmosphere for gas exchange, and for the removal of plant metabolite waste. Next, we will proceed with composition of culture media. It is composed of four main elements, which is inorganic nutrient, organic nutrient, other media substance, and pH. For the inorganic nutrients, it includes macronutrients like nitrogen and phosphorus, and also micronutrients like boron and copper. For the organic nutrients, it includes vitamins like vitamin B1 and B6. For the other media substance, it's like protein hydrolysates and yeast extracts, for example like fruits and banana. I will proceed with explaining the inorganic nutrients. Mineral elements play a very important role in the growth of plant functions of nutrients in plant growth. Under inorganic nutrient, there are macronutrients. Macronutrients is element required in the life of a plant greater than 0.5 mole and referred as macronutrients. It includes 6 major elements as nitrogen, potassium, phosphorus, calcium, magnesium, and sulfur.
Plant tissue and organs are grown in vitro on artificial media that provides beneficial nutrients for growth. The culture medium used greatly affect the success of plant tissue culture as a means of plant propagation. Therefore, for an intact plant to grow healthily, one of the crucial elements in composition of culture media is inorganic nutrients. So, what is inorganic nutrients? Inorganic nutrients are minerals that play an important role in plant growth. They are divided into macronutrients and micronutrients. I am going to present about micronutrients. Micronutrients are the minor plant nutrients. It is also called as trace elements. They have roles in the functioning of the genetic apparatus, and several are involved with the activity of growth substances. Without micronutrients, a medium does not encourage healthy growth and the plant will exhibit many deficiencies. This is because micronutrients play a role as the growth substances function. The essential micronutrients for the growth of plant tissue culture are molybdenum, iron, manganese, boron, zinc, chlorine, and copper. They are usually needed in small quantity, which is less than 0.5 millimole per liter. Now I'm going to explain about the function of each element. The first element is iron. Iron promotes chlorophyll formation. It is also present in many enzyme mechanisms to operate respiratory system of the cells. The next element is zinc. Zinc promotes the growth hormones such as auxin. It is also needed to form seeds and grains. In addition, it is necessary for starch formation. The next element is manganese. Manganese acts as activator of enzyme reactions such as oxidation and reduction. It also has direct influence on sunlight conversion in the chloroplast, which means manganese is an aid in chlorophyll photosynthesis. Next is boron. Boron promotes maturity of the plant and therefore it is essential for seed and cell wall formation. Boron also involves in synthesizing protein. The next element is copper. Copper improves flavor of fruits and vegetables. Copper also portrays a major function in photosynthesis and reproduction stage. The next micronutrient is molybdenum. Molybdenum plays an important role in plant nodulation. It is also essential for nitrogen fixation and assimilation. The last micronutrient is chlorine. Chlorine regulates turgor pressure and is important in drought resistance regarding the stomata gate cells. It also encounters ion to positively charged cations in the cell. As I said earlier, there are 3 types of nutrients needed for plant tissue culture media. The composition of plant tissue culture media consists of inorganic nutrients, organic nutrients, and others. Now, I'm going to talk about organic nutrients. Organic nutrients are molecules that contain carbon and oxygen. It includes nitrogen sources, amino acid, vitamins, carbon sources, and growth hormones and regulators. I am going to present on nitrogen sources. Nitrogen is the major component of all plant tissue culture media. It helps in synthesizing complex organic molecules. In plant nutrient medium, nitric ions, ammonium salt, amino acids, and complex organic compounds supply nitrogen. Hence, nitrogen in the form of ammonium and nitrate are the dominant mineral nutrients in most plant tissue culture formulations. Other than nitrogen source, organic nutrients also include vitamins, amino acids, carbon source, and growth hormones or regulators. These are the types of vitamins in organic nutrients. First, thiamine or vitamin B1. Thiamine, along with cytokinin, helps in the growth of callus and rooting in tissue culture. It can also facilitate secondary metabolites production in certain plants. Next, nicotic acid or niacin vitamin B3, pyridoxine, vitamin B6, and calcium pentosanate, vitamin B3, are needed to help improve the growth of the tissue culture material. Myoinositol in a phosphate form is part of the B complex of the cell membranes and organelles. It is not an essential nutrient for growth. However, it can benefit the plant in many other biosynthetic pathways. Cyanocobalamin, vitamin B12, riboflavin, vitamin B1, folic acid, vitamin M, biotin, vitamin H, p-amino benzoic acid or PABA, ascorbic acid, vitamin C, and alpha-tocopherol, vitamin E, are usually added in special cases. However, their exact role in tissue culture is yet to be known. The third organic nutrient is amino acids. Cells in certain cultured plants can synthesize all non-essential amino acids. However, can we add amino acids alone into the cell culture? The answer is no. If only amino acids are added into the cell culture, the plant cell growth can be inhibited. Therefore, we only use these in culture media, which is amino acid mixtures, for example, casein hydrolysate, L-glutamine, L-asparagine, arginine, methionine, and adenine. Other than that, tyrosine has also been used in cell cultures to stimulate morphogenesis. However, tyrosine can only be used in an agar medium. Adding adenine sulfate may also stimulate cell growth and enhance the shoot formation of cell. Next organic nutrient is the carbon source. Most plants are non-autotrophic, which means they are unable to produce their own food. Therefore, they only rely on external source of food, which is carbohydrates, as their main source of energy. The most commonly used carbon source is sucrose, for about 2 to 5%. Generally, the dicotyledonous roots always grow better in the presence of sucrose. Meanwhile, glucose and fructose are used for cell growth. Monocots usually grow best in the presence of glucose. Maltose and raffinose are used, but only in some cases. Other carbohydrates such as mannose, sorbitol, pentoses, sugar alcohol, glycols, hexose, uronic acid, lactose, galactose, potato starch, grain starch, and even glycosides can be used depending on the condition of the plant cell culture. The last organic nutrients needed in plant cell culture are the growth hormones, or regulators. The plant tissue culture highly depends on the amount of growth hormones added into the medium. Therefore, the amount of hormones may vary for each plant. There are 5 classes of naturally occurring plant hormones, which are auxins, ethylene, abscisic acid, cytokinins, and gibberellins. Other plant hormones, such as polyamides, jasmonates, salicylates, are also used, but it depends on the condition of the plant culture.
Growth regulators are chemicals and signal molecules that affect the growth, formation, development, or division of plant cells. There are 5 classes of growth regulators, which are auxin, cytokinin, gibberellin, ethylene, and abscisic acid.
First is auxin.
It will promote roots growth and cell division. In organized tissue, auxins will cause root formation, delaying leaf senescence, fruit ripening, and use in embryogenesis. Commonly used natural auxin is indole 3-acetic acid, while synthetic auxin are 1-naphthalene acetic acid and 2,4-dichlorophenol acetic acid. Second is cytokinin. It will promote shoot growth and cell division. It is useful in culture for stimulation of cell division, release of lateral bud dormancy, and induced adventitious bud formation. Cell division is regulated by the joint actions of auxins and cytokinins. Auxins affect DNA replication, while cytokinins exert some control over the events leading to mitosis. In intact plants, cytokinin promote lateral bud growth and leaf expansion. It also will promote chlorophyll synthesis and enhance chloroplast development. Zeatin and 2-IP or 6-gamma-gamma dimethylaminopurin are naturally occurring cytokinins, while synthetic are kinetin and benzyladenine. Third are gibberellin. It will promote flowering, seed germination, and stem or shoot elongation. There are over 20 known gibberellin, and GA3 is used to increase the shoot elongation. Culture callus cells will synthesize their own gibberellin. Next is ethylene. It is a gaseous hormone synthesized in cultured cells, fungi, and bacteria. Ethylene gas promotes fruit ripening, senescence, and leaf abscission. At higher concentration, the gas decreases cell elongation but increases cell expansion. The role of ethylene can be difficult to understand because its effects vary in development stage and because low concentration can promote or sometimes inhibit a process, while higher levels have the opposite effect. Ethylene is synthesized from methionine. Auxin will stimulate the production of ethylene, but the physiological significance of ethylene in tissue culture quite unclear. Lastly is abscisic acid or ABA. It is plant stress hormone and it inhibits auxin. It is naturally produced in plant tissue by the cleavage of xanthophyll. ABA is often regarded as being an inhibitor as it maintains bud and seed dormancy, inhibits auxin-promoted cell wall acidification loosening, and slow cell elongation. ABA plays a key role in closing of stomata aperture and abscission of leaf. It also controls water and iron uptake by roots.
So we have learned that a composition of a culture media consists of inorganic nutrients, organic nutrients. So now let us see what other substances may contain in a culture media. A culture media may consist of protein hydrolysates, yeast extracts, fruit extracts, coconut milk, solidifying agents, ion sources like EDTA, and antibiotics. Other media substances that also can be found in the media are phenolic compounds like fluoroglycinol, which stimulates rooting of shoot sections, and detoxifying agents like activated charcoal, which will detoxify waste from plant tissues and impurities. However, addition of activated charcoal to certain cultures like tobacco and soybean is found to be inhibitors due to the absorption of growth stimulants such as phytohormones. Solidifying agents. For preparing semi-solid or solid tissue culture media, solidifying or gelling agents are required. Solidifying agents extend support to tissues growing in the static conditions. Solid media are often preferred because it improves oxygen supply and supports culture growth. The commonly used substance is agar, and others are alginate, carrageenan, gelatin, starch, silica gel, hydroxyl ethyl cellulose, and polyacrylamide. So agar is a polysaccharide obtained from seaweeds
is most commonly used as gelling agent with concentration of 0.5 to 1%. Its properties, where it does not react with media constituents, not digested by plant enzymes, and is stable at culture temperature, make it why it is the most commonly used gelling agents. Another famous solidifying agents are gelatin. However, it is not commonly used in media because it needs high concentration, which is 10%, but with a limited success. Gelatin also melts at low temperature, which is 25 degrees Celsius, and consequently, the gelling property is lost. Next, antibiotics. Antibiotics are used for prevention of excessive contamination in the culture medium. Generally, fungicides and bactericides are used in culture medium but are not being very useful because they can be toxic to the egg plant and the contaminant sometimes reappears as soon as they are removed. Commonly used antibiotics are carbapenicillin and augmentin. However, the addition of antibiotics to the medium is avoided as far as possible as they have an inhibitory influence on the cell growth. The pH of the medium also plays the important role. The optimum pH for most tissue cultures range between 5.6 to 6.0 before autoclaving the culture medium, and the pH generally falls by 0.3 to 0.5 units after autoclaving. At a pH higher than 7 and lower than 4.5, the plant cells stop growing in cultures. If the pH falls during the plant tissue culture, then fresh medium should be prepared. In general, pH above 6 gives the medium hard appearance, while pH below 5 does not allow gelling of the medium. So that's why pH also plays important role in the media.